First year we have learned how the fixed dome digester is working, and we are going to try to modify design, and we're going to have this fuel digester, and we are going to have a lab digester in a small and lab scale in order to test different approaches. One is trying to separate two important functions inside the digester, that is hydrolysis and methanogenesis, and the other thing is we need to improve the ratio between microorganism manure in order to increase the gas production.
The water sample, and we look at the temperature of the digester and ambient air, outside temperature as well. The methanogens, the bacteria that are actually making them, the methane that are breaking down the organics from the manure into acetic acid and then methane are very temperature dependent. So the temperature of the digester is one of the most important variables that we try to control and monitor. And what's important there is the quality of the water, looking at how much what's important, how much organics, what type of organics are in the water, comparing that to the feedstock gives us an idea of how fast the degradation processes are occurring inside the digester. And then what's also important is not just water quality, but actually looking at the diversity and the types of microbial species that are inside the digester, because that'll tell us how well the digester is performing. It'll also tell us how many methanogens we have relative to acetogens, and that'll tell us how well the digester is performing, and it'll help us learn how to design it better and manage it better so we can make more biogas. This is some really stinky biogas, probably composition. Based on our previous results, we probably have about 25% methane, maybe 27, and the rest is mostly CO2. And that's actually not where we'd like it. Most digesters have a methane percent that's around 60–65%. And so we're trying to figure out exactly why, and there's a few possibilities. One is that we didn't start this until October when it was already getting cold out here, so the microbial community might not be fully evolved yet. That's one possibility. The pH is a little bit low. The pH is around 6.2 right now. Ideally, it would be around 7. Another possibility is there could be ammonia inhibition, because we are using manure. One of the problems with manure digesters is ammonia inhibition. So we could have ammonia in the digester. We're starting to collect some water samples to look at that. So it could be any of those problems. It could be a combination of those three. It could be something else. But it is starting to perform a lot better as it's getting warmer here. We're getting a lot more biogas, and the percent is starting to creep up a little bit. It was around 25% a few weeks ago. Now we're up to about 28%. So, but it still smells really bad.